Okay, transgenic organisms, um, like transgenic corn or genetically modified corn, how are they created? And most of the time, they're created by using a vector or a vehicle for transmission of a gene from one species to another, and that transformation or that transpiration, that means of transpiration, I finally said it, is usually bacteria. Now, the process of putting the two genes together is a type of technology referred to as recombinant DNA technology. And that's the joining of DNA from two different species. So you're recombining the DNA because, or based on the fact that DNA is a universal code. So all organisms, no matter what it is, have A's and T's and C's and G's that follow the DNA base pair rules. You can do a little clicking here. So in order for this to happen, the bacteria have a single circular piece of DNA called a plasmid that's going to be used to transport the gene from the one species to the new species. So there are enzymes involved in cutting genes, and there are enzymes involved in binding, sealing them together. So this plasmid transformation, which are those single circular pieces of DNA, what can they be used for? So we're going to kind of look at some of the uses, and then we'll roll back to look at some illustrations of how they happen. So DNA recombinant technology, or DNA cloning, is the process of making many copies of a specific DNA sequence in order to make lots of a protein. So whatever purpose that you have, you want to be able to make mass quantities of that. The copies are typically made in bacteria by introducing foreign DNA into the plasmid, and that's called transformation. So some examples, and there are a gazillion examples, the production of insulin for diabetes. So insulin helps to regulate your blood sugar levels, and some people have issues with the production of insulin. So therefore their blood sugar might get too high. So having that, and having mass quantities of it made quickly and cheaply is a benefit for people. And that's usually done in organisms like cows or pigs. Now, it's still going to be human insulin, but the process of making this human insulin be done in cows or pigs. And then this TPA, which sounds like, I don't know, the Toilet Paper Association, but what it stands for is tissue plasminogen activator. And what it is something to help people who may clot too frequently. So when they bleed, their body might clot more than what it should. And this is something that can be provided to them to prevent them from over collection of what are called thrombocytes. When you bleed, these platelets, these thrombocytes come together to stop the bleeding, to prevent that from happening too much, to dissolve those potential blood clots. So we're going to look a little bit at traditional cloning. So, one of those best parts of talking about genetic engineering or biotechnology is looking at cloning. So if you can manage to make this transgenic organism that's going to be able to make human insulin, or these factors to prevent somebody from having blood clots that could potentially kill them through cloning, you could make a gazillion of them. So I will catch you on the other side of this cloning video.